weight of the bacteria is going to be carbon. So we need to have a carbon source: glucose, carbohydrates, proteins, and amino acids. You could also feed oil or glycerol or methanol or other organic chemicals. Okay, organic meaning it has carbon. Then we need a nitrogen source, which is about 12% of the bacteria composition. Most bacteria can use inorganic nitrogen, which is NH3. There's also organic nitrogen, which would be proteins. Okay, proteins and amino acids are considered organic nitrogen. So most bugs can use most amino acids. They maybe can't use all of them. It depends on the bug. But typically they can take in the amino acid and they can convert it to other types of amino acids. They can do the same thing with ammonia. Most bacteria are able to just take in inorganic nitrogen and use that to produce amino acids, which are used to produce proteins. There's also nitrogen in nucleotides. So they need the nitrogen also for their DNA. Okay. And there's other macronutrients. Okay. Phosphorus is required for nucleotide synthesis and ATP. Sulfur is used in cysteine. It's an amino acid with this sulfur in it that makes disulfide bonds. Methionine has sulfur in it. Some vitamins and precursors may have phosphorus or sulfur in them. And then there's also potassium and magnesium. These are all considered macronutrients, so there's more of them in it. And there's also micronutrients, which would be like manganese and cobalt and iron and other ones that, and all those we don't need to worry. We're not going to worry about it because it's going to be in our LB medium, in the yeast extract, so we're not going to have to worry about including these micronutrients. And there's also vitamins and other growth factors in the yeast extract and the tryptome that's in the LB medium, so we don't need to worry about these either. But most bacteria need some, one vitamin. Not necessarily, they may be able to live on it purely. If they can synthesize all of their own, then they don't need them. But that's how it is. This is a typical, in the past, we made this media in this class. And I think the students here, I had prepared the trace metals ahead of time, but I just decided we're not going to do it. Okay, so the disadvantages of using complex media are that it comes from natural sources, right? Tryptone, I believe, is a digest of milk. That's what it smells like. And then yeast extract. So these start from natural sources. So obviously the composition of them is going to vary. And so you're going to get variability, and it can cost more, but it may not cost more. There are some complex media which are really low cost, and they provide nitrogen, proteins in a low-cost way that can actually really boost the productivity of your process. So it depends on what your outcome is that you want to try to achieve. You may be better off with complex media. It certainly is easier to use. Although sometimes it requires maceration. We don't have to worry about that, but you might need to grind your stuff. Okay, and then so advantages of complex media is that it contains some components that may be synthesized by the cells, and then the cells don't need to synthesize them, so that's really actually a major advantage. So you might result in higher growth rate, higher productivity, and as a result, you get cost savings from it, and higher density of your culture, and the effort to include all these ingredients. So it can really be advantageous to find a cheap source of a good complex media for your process. So in our process, we are going to be growing E. coli that expresses green fluorescent protein. And we have a plasmid that confers kanamycin resistance. So we are going to add kanamycin antibiotic to our process post-sterile addition. We're going to add that antibiotic. If you add it before the sterilization process, you will probably destroy it. So it's important to wait until before you inoculate to add the antibiotic. So in terms of the inducer, the gene for green fluorescent protein is downstream of the T7 promoter, which is a bacteriophage. I hope you know what a bacteriophage is. But it expresses a lot of protein, And in order for that to express a protein, it needs the T7 RNA polymerase. And this bacteria has the gene for the T7 RNA polymerase downstream of a LAC promoter, which means that lactose, or I wrote the word down here, isopropyl beta-D-thiogalactoside, which is a lactose mimic, and it won't be consumed. So if you put some of this in there, it stays there. It doesn't get eaten up. Okay, whatever it is, this produces in our system without adding any inducer. It must be in the either in the tryptone or the yeast extract. There must be something that causes this induction to happen. So we don't need to worry about it. That's all it is. Okay. So the media for our fermentation will be, here it is, per liter. We're going to make 2 liters, so double these values. 10 grams of tryptone, 5 grams of yeast extract, 10 grams of salt. Okay, we prepare 2 liters of that. We're going to autoclave the bioreactor with 2 liters of this stuff. And then right before we run, right before we inoculate, we will add some glucose solution. And we'll make, we want to have a total of 5 grams per liter of glucose. Okay, and that glucose is going to have, be in a solution of water and glucose. Note also that dextrose and glucose are the same thing. Okay, so now we're going to talk a little bit about glucose. Okay, so we like to use a very highly concentrated solution of glucose because we don't want to add useless water to our process, because it just fills up the bioreactor that we don't need to do. So we're going to make a concentration of like a glucose syrup, right? Okay, so these are some points about starch. We're just going to use straight glucose, and that's kind of interesting. Okay. We don't need to worry about this either because we're just going to use LB media. And one thing to point out is that if the media is above pH 5, you don't want to autoclave the glucose together in the media because you will caramelize it. Okay. All right. So one thing to note is that we have a nice, like it's just basically a big pressure cooker in the lab that we're going to use to autoclave the bioreactor. And so we are going to do that carefully. We can prepare the bioreactor, and I think I have described here how to prepare it. And then we're going to put it right down.